Please make Dr. Michael Cowell welcome.
Thank you. It's a real pleasure to be here, and I want to take the opportunity to thank the organizers for inviting me to spend 2 wonderful days here with you. I've been really deeply impressed with the wonderful engagement that the Australian FASD community has. It's really very impressive, and I've been very touched by the many sort of personal stories that I've heard over those 2 days, and especially seeing that our work, as you'll see in a second, is rather molecular, is very meaningful and to me. And I'm honored to discuss today our work with you, and particularly because it's such a diverse audience. So what are we going to do? I can't see it on here. This is not working. Okay, I'm going to get a stiff neck. So what I'd like to do today is just to spend a few minutes continuing from where Jeff started to discuss with you how it has become that epigenetics becomes such a crucial player at the interface between the genome and the environment. And then I've heard a lot of talk about environments broadly speaking the last couple of days, so I want to share with you some of the work we have been doing over the years to show that there are indeed variety of environments, including poverty and family environments, get under the skin to affect our biology at the level of the DNA. And I'm going to move on to discuss work we've been doing on fetal alcohol spectrum disorder. And collectively, what I'd love for you to take away from this is that indeed epigenetics might be a key player at the interface of nature and nurture, but also that there are many different aspects, many different environments that actually affect the epigenome. So Jeff already alluded briefly to what epigenetics is. You can see the most common definition here on the screen. I actually hate definitions. I'm German, and we're supposed to love definitions. I hate them, which is probably why I live in Canada. So the particular aspect of epigenetics that we're really focusing on though is the social environment. And really the idea that there are experiences, environments, and particularly those in early life can get under our skin to affect our biology. And the kind of paradigm that we're working under is that these experiences, they affect the biological pathways by getting on our skin, and they drill all the way onto the level into the DNA, into the nucleus of a cell. Of course, this is not a one-way road because you can then easily imagine this to go the other way as well. So when we think of epigenetics, we really just need to understand that little DNA piece there, and we need to understand that we have 25,000 genes. And with all due respect to my molecular colleagues, all you really need to know is a gene is essentially a light bulb. Okay, so if you know, a light bulb can be either totally on or totally off. Or if you live in a fancy house, you have a dimmer, and you can put them somewhere in between. And the epigenetics, in particular, the kind of DNA methylation that Jeff alluded to, if it's in the gene regulatory region of genes, it's really just a dimmer. So that's all you need to know about epigenetics to get your certificate as an epigeneticist. Now, how does that look in a particular situation? So let's say you have your favorite gene. In this particular situation, we have no methylation, but and the light bulb is all the way on. Now we start to methylate. Methylation is just really a tiny little chemical tag consisting of some carbons and hydrogens. And now we start to methylate. And you see how we methylate. We push that dimmer down. We push it further down. And ultimately, we have fully methylated this gene. And we have turned the light bulb off. That's as simple as it is. Now, how does it look in real life? Well, if you look at these 5 young mice, is there anyone in the room who thinks that they look exactly the same? Good. Otherwise, the optometrist has a work to do. So we all agree, I think, that their coat color is quite different. But what I'm going to tell you is that they're genetically actually identical. So it's not that they have different genes. The only reason that they have different coat colors is that their mom was fed a different diet during pregnancy. And this particular coat color is determined by one gene. And on the mice on the left, this particular gene is totally on. The dimmer is all the way up. We have no methylation. But as we go to the mice on the right, we start to methylate and methylate this gene. We turn that light bulb off, and now you have a different coat color. Okay? And again, this is solely dependent on the nutrition of the mom during pregnancy. And there are a number of compounds that have been shown to affect this kind of circuitry. Now, of course, most people really don't care whether they have yellow or brown mice in their house, as long as they don't eat the cheese. But let's just wait what happens to these mice when they grow up. And keep in mind, again, they're genetically identical, because those people in the basements that grow the mice, that's what they do. The only difference was that their mom was fed a different diet during pregnancy. So you see the powerful impact of this epigenetics. You see that there was a change in these stimulus switches that was dependent on the mom's diet during pregnancy. It got under the skin of these mice, it stuck with them for the rest of their life to lead to these vastly different health outcomes. This touches on all kinds of things, including mental health, but also, as you can see, metabolic health and the like. Now, this is very powerful. And since, as Jeff alluded to, this has been actually shown to be true in human as well, as we can see here in the Dutch famine, where we have these associations of the maternal diet and the caloric intake with the dimmer switches in the offspring. There's also prenatal smoking that has been shown to affect this circuitry, and a number of other compounds. Now, the field really has exploded some 10 years ago with seminal work by Michael Meany from McGill University in Canada, who showed that actually the quality of the maternal care also can affect these dimmers. And so this work really has brought this entire field to the attention of the public. I'm going to show you a little bit of data related to this. Now, of course, the closest we can come to these cute mice are identical twins that Jeff alluded to. And so you see those two cuties here. They're identical twins, but obviously they don't quite are the same. And yet, if people look at their epigenome, it turns out that the epigenome is very similar early in life, and it becomes divergent later in life, in particular if these twins had been separated early in life. In turn, these kind of modifications then are also associated with a host of health outcomes, including mental health, as shown primarily by Jonathan Mill from the UK. Now, taking all of this together, my team really focuses on the role of epigenetics in child health, broadly speaking, and really at the interface of nature, which is our DNA, and nurture, which is our environment. And ultimately, what we're trying to understand with this integrative view is to how can we provide the best and most healthy starts to our children, and how can we potentially prevent disease. And we had the honor of working on a number of interesting projects and over the years that I think are highly relevant for this aspect to really understand how experiences get under skin. And as you might know, there are very few experiences that are more powerful than socioeconomic status. And so we've been focusing on that in a suite of work to understand, does it matter whether you grew up in a kind of dwelling on the left or in the mansion on the right? We've also been focusing on the family unit. Does it matter whether you have a sort of family with the nuclear family with doggy woof woof, or the family where the parents constantly yell at each other? And ultimately, how does that then turn around to affect childhood temperament and behavior and trajectories? And this is really sort of rooted in this idea that there are developmental origins of health and disease that can trace all the way back, as Jeff so beautifully showed to you, to the womb. But we're focusing on these early years of life and trying to understand how these experiences get under the skin. And of course, the idea is that that happens by changing these dimmer switches and the light bulbs. And so in a suite of studies, we really focused on adults, trying to test whether we can see a vestige of early life poverty. And this was published some years ago, but what we did, we essentially had two groups of adults. They were all healthy adults that were separated by whether they grew up the first 5 years in low versus high socioeconomic status, but were mixed in adulthood, so that whatever we measured was not confounded by the adult socioeconomic status. And then we asked a number of questions. We first asked, are these light bulbs different? And indeed, as you can see, they're actually different. And without going into too much detail, they're not different in a random way, but they're different in a way that suggests that the folks with that grew up in low socioeconomic status actually have an elevated immune response, even as adults, that can be traced back to their socioeconomic status in early life. And lo and behold, when we then directly test their immune function, you can see that on the top right, in fact, the prediction came true that these people had constantly a higher engagement of their innate immune pathways. Further, these folks also had a consistently higher cortisol output, so their arousal level, their stress level, if you wish, was higher throughout the day. And again, all of this can be traced back to their socioeconomic status early in life and not their current socioeconomic status. Further, we can also show, as seen by these highly skewed distributions of these bars, what you want to see is things going to the left, that there is an association of early life socioeconomic status with these dimmers in these people. Interestingly, at least in this study and some others that we've done by now, there's actually no association of your current socioeconomic status with these dimmers, suggesting again that collectively the early life poverty gets under the skin to set these dimmers in a persistent way that lasted all the way into adulthood, that change the light bulb setting, and that change the function of the immune system and the stress reactivity of people. Now, socioeconomic status, of course, as you know, is a very highly complex construct, and so we're constantly trying to sort of tease it apart even more. In a paper we recently published together with Tom Boyce and Nikki Bush in a cohort from the San Francisco area, we were actually able to take socioeconomic status apart into three components, all of which, actually, as you can see by these, again, skewed distribution of these bars to the left, and these little dots that are either blue or red, all of those are associated with either income per dependent, parental education, or family adversity, suggesting that each of those has a distinct signature leaves a distinct signature into the epigenome, in this case, of these children that are between 8 and 12 years old. Now, we also have been then looking more closely, motivated by the fact that we wanted to see whether we can potentially use this work to provide some recommendations as to what families can do. We have been interested whether there is indeed also a signature of the family unit that goes beyond just adversity. And in work that we published last year, we actually shown that these dimmers can be traced back to whether a neonate actually receives a lot of caretaker interaction versus very little caretaker interaction. And this work, of course, as you can imagine, caught quite a bit of public attention. So the Vancouver newspaper said cuddling babies or not affects infants' gene expression. Scary stuff. But we'll come back to this later. Now, how can we apply all of this then collectively to really get a bigger picture? Well, we can do that by adding more and more studies, and that's exactly what we have done. And so we have published studies that show kids growing up in orphanages in Eastern Europe being adopted to the United States. We see these signatures of dimmers. We see these signatures of the immune system. We have done studies in the Philippines where we can show that in young adults, again, there is a signature of both their social but also their physical environment in their white blood cells. And so collectively, really a picture is emerging by which these epigenetic modifications early in life do get under are being sculpted by a number of different environments. And together with my mentor and friend Tom Boyce, we have then put this recently into a more comprehensive review, trying to sort of understand how this can be really serve at the synapse of the gene environment interplay to help our children have a rocket start to life. Now, how can we apply all of this to FASD? As you know, much better than I do, FASD is a multifaceted syndrome. In Canada, as we heard yesterday, the incidence is between 2 and 5%. And so we have then put this epigenetic biology to the test in the context of FASD. And we've done this under the umbrella of the Kids Brain Health Network, formerly NeuroDevNet, a Canadian organization that is dedicated to understanding neurodevelopmental disorders from the basic biology all the way to the advocacy. And really, what we try to do with this work is twofold. We try to ask the question whether epigenetics can tell us something about the biology of the disease, of the disorder, and perhaps more tangibly, whether epigenetics can serve as a new biomarker for better, earlier, or more sophisticated diagnosis, or just another tool in the toolkit. And I'm happy to say that I think the early results would suggest that both of this could be the case. So we've been building this on 2 Canadian cohorts, the NeuroDevNet cohorts, FASD cohorts. The bigger one to the left is a pan-Canadian cohort You can see 110 cases and around 96 controls, and 110 cases are distributed across the range of FASD, as you can see. So it's versus the second cohort is smaller. It's ARP Chudley's cohort. Many of you I know probably know ARP very well. It's the Manitoba cohort, and it's smaller, but it's only children with FASD. So using the first cohort, we asked again the question, are there differences between the case and controls in these demo switches? And long and behold, as you can see here, there are lots of blue dots, so you just want to focus on the blue dots, and you want to focus on blue dots that go far away from the middle, and they go high up. So this is called a volcano plot. And really, because what is true in most up and far is really the most meaningful associations. You can see there are roughly 650 or so CBGs in more than 400 different genes that are associated with fetal alcohol spectrum disorder in this first cohort. When we look more closely at those, we can see that there are actually a number of them that make biological sense, even though this is in cheek swabs, including components such as DLD4 that are part of the dopaminergic system. And we can come back to DLD4 in, I believe, 3 slides, so hold your questions. Now, we can also look beyond these individual dots and look for broader regions. When we do that with the bioinformatic tools that are available, we can identify epigenetic neighborhoods that differ between kids with FASD and those the controls. And again, we see changes in genes that make a lot of biological sense. We see changes in genes that are related to the immune response. And as you know, individuals with FASD tend to have compromised immune function and higher incidence of arthritis and the like. We also see genes that are associated with autism spectrum disorder, perhaps not entirely surprisingly. And when we then do even more sophisticated analysis, then I'm going to spare you, because I barely understand it myself, to look at sort of network analysis, we can see that there is indeed an overlap potentially with other neurodevelopmental processes in disorder, again, including autism, anxiety, and epilepsy. Now, of course, in our world, you're only worth as much as you can replicate, so this is why we turned, after this sort of initial promising work to NeuroDefinite2, ARP Chudley's cohort, to ask to what extent can we actually replicate this. And in work that Alex Lucy, a very talented former PhD student, published just earlier this year, you can see that we can replicate roughly 180 of those 650 CBGs. And those are the ones, if you look at the left, that are red. You can see that many others also go in the same direction, but they do not pass our fairly rigorous statistical threshold. As Jeff pointed out, this is all done by very sophisticated technology called Array, and so often what we try to do to use a different technology, and we use what's called pyrosequencing to look whether this actually holds up and is not just an artifact of the technology. And as you can see here, for this gene that's involved in brain development and calcium channel and substance-related disorders and epilepsy, you can see that both technologies give a very similar pattern of association with, between FASD and controls. But the reason I really put this up is that you can also see, if you take a single individual on this plot, on the number C on the bottom, if you take a single individual just based on this one gene, you actually cannot tell whether this person would be part of the control group or whether it would be part of the FASD cohort. And so how do we get around this? Well, we get around this by using the black box and the magic word that everybody loves these days, machine learning. So what Alex did, he used the first cohort, the NeuroDefinite cohort 1, and had this DNA, the signature of the FASD that I showed to you, and then he used machine learning, which you can talk about later, to essentially come up with a predictor for FASD that's not just based on one single of these dimmer switches, but uses some fancy thing called neural nets to identify patterns in these data. And when he does that, he then tests this in the FASD cohort, the second one, and then in the negative control, which is the autism spectrum disorder cohort from publicly available. And when you look at these curves, these receive operating characteristics curves, you see actually we're getting somewhere. We definitely, if there was no predictive value, these blue and the gray things would just go diagonally up the middle. But you can see that there's sort of this nice kind of shape that we're looking for. It's not perfect. When we then start mixing this and putting it right to the test in the NeuroDefinite2 cohort, you can see that the predictor actually predicts 18 of the 24 FASD cases. It misses 6 and also mislabels 2 controls as FASD. So it's not perfect yet, but I think it's a very promising beginning that we can use these kind of technologies and these kind of approaches to understand, to come up with biomarker and a predictor for fetal alcohol spectrum disorder that hopefully can be added to the toolbox. Now, we also then wanted to understand the biology just a wee bit more, and there's very little you can do in the human biology other than measuring these associations that I talked about. And so we turned, together with my dear friend Joanne Weinberg, we turned to the rat. And Alex just published a paper, actually, it's not published, it's just accepted this week, where he uses Joanne Weinberg's paradigm of 3 different feeding patterns prenatally with the rat control, nutritional control called PAIR-FED, and the prenatal alcohol exposure. And there's a long story to this, but Alex was essentially then wondering how it is dimmer switch is looking in the brain, in the hypothalamus, which is a key brain area involved in FASD and in the stress response in these rats. And he identified 30 different regions across the rat development that were differentially methylated and could be associated with the exposure to prenatal alcohol. And then, of course, the key question becomes, well, can we actually draw something about this biology and compare it to the human data? And you see it from the title, it's a little bit like comparing apples and oranges. So the good message is, we humans aren't rats. And of course, our brain and our cheek cells are quite different. But nonetheless, there was actually one particular gene among all of these different dimmer switches that Alex identified that was different, that was common between those 2 paradigms. Any wild guesses? Liz? Well, I told you 3 slides ago, DLD4. So the DLD4 gene, which is involved in the dopamine pathway, as I said earlier, is actually, you can see that here again in these pyrosequencing patterns, it's very differently methylated. These dimmer switches are very different between the exposed rats, which are in red at the top, and the blue rats, which are the control rats. And this is true across the entire span of development. And if you look at the human, again, it's also differentially methylated. There are many caveats to this. It's not exactly the same region. It goes different directions. But nonetheless, I think it's very telling, very exciting. And it's even more exciting when we look at a paper that lovely Australian folks, some of which are in the room, published that also looked at perinatal maternal alcohol consumption and DLD4 methylation. So maybe it's a sign that we should work closely together to explore that. With that, I want to just sort of close and really share with you some of my thoughts on all of this. So first of all, and I sort of divided it up in different pieces. First of all, I want to make sure that you understand this is all correlation, and it does not imply causality. That's a very important point. Secondly, I also want to highlight that by and large, with some exceptions, these DNA methylation changes aren't necessarily bad or good. They can mean all kinds of different things, or they can mean nothing, and can be just a biomarker. I also want to just follow up and just highlight again that we have a gap between these group averages and individual predictions that we're trying to close with these machine learning approaches, but the gap exists still very much. So again, this is an important aspect. On the more technical level, of course, our cohorts were such that we can't really distinguish a signature between PAE and versus FASD. And as you probably can tell from all the different environments that are shown to be associated with DNA methylation, many different confounding factors contribute to shaping the epigenome, some of which might be overlapping and some of which might not. There is also this issue of tissue-specific versus tissue-agnostic measures. And then, of course, what we have not yet touched upon is that there might well be a genetic underpinning to all of this. But what I'm really most excited about is really this knowledge translation, the opportunities that all of this offers. And as you probably can tell, it's a fairly complex biological concept. And the key point that I think I really love to discuss further is this idea that there could be an epigenetic risk messaging. And that the idea that there could be a biological method determinism, I want to strongly argue that this work is actually not the case. And I'm very curious to learn when you think about this work, whether it actually increases or decreases FASD stigmatization. And we're actually starting to have focus groups in Vancouver now that address these questions that include a number of different stakeholders. But collectively, I hope that I've shared with you my excitement about early life experiences going all the way back to prenatal, and their way getting under the skin via the modifications of these demos, the epigenome, the interaction with the light bulbs to affect phenotypes, broadly speaking. So thank you so very much.